So now our next speaker is Dr. Simerjeet Kaur ma'am. And Dr. Simerjeet ma'am is a principal agronomist at Department of Agronomy, Punjab Agriculture University, Ludhiana. She has earlier served as assistant professor at KVK Kapurthala and agronomist and senior agronomist at PAU. Dr. Kaur has more than 2 decades of research and teaching experience with major contribution in weed management and conservation agriculture based cropping system. She served in All India Coordinated Research Program on weed management for a decades, and under her guidance only, her center recognized the Best Center Award in 2016. She has developed more than 30 crop production and production technologies. She has published nearly 100 research papers. Out of that, nearly 28 research papers has published with high impact factor. She has authored of 6 books, 11 book chapters, 16 edited books, and many more. She has guided more than 10 students, and she has taught more than 25 courses in agronomy and including weed science. Dr. Kaur has played a key editorial role in the serving as the co-editor-in-chief in Indian Journal of Agronomy, associate chief editor of the Indian Journal of Weed Science, Crop Sciences, Journal of Agriculture Sciences, Cambridge, and Frontiers in Agronomy. She has presented more than 50 papers in national and international journal, international symposia and conferences, and she has received the advanced training in the USA. Her contribution has recognized with the PAU Appreciation Certificate and Fellow of Indian Society of Agronomy and Indian Society of Weed Science. We are honored to have ma'am for today's talk, and very important talk, that is management of herbicide resistance weed in rice wheat cropping system. So ma'am, we are welcoming you again, and now the screen is yours, ma'am.
Good afternoon, everyone. Am I audible, Dr. Jeetendra?
Yes, ma'am, you are very much audible.
Thank you, Dr. Jeetendra, for a nice introduction. So I'm sharing the screen first. So I think it is visible.
Yes, ma'am, it's visible.
Right. So welcome everyone to the talk, which is management of herbicide-resistant weeds in rice wheat cropping system. Basically, everyone know that chemical weed control is the easiest, simplest, effective, cheapest method. And I was listening to the previous talk by Dr. Shobha. There are many problems of residues. Yeah, they are. This is a very good weed control method. Farmers adopt it very eagerly, but then there are some problems due to inadvertent or not judicious use. When we are not doing that, then there can be problems, and one of the main problem is evolution of herbicide resistance in the weeds. First of all, overall scenario, if we see that as compared to the world, India is using only 16% of herbicide as compared to other pesticide. Like I have given the data, herbicide in India is only 6,334, whereas we are having insecticide at number 1. And in contrast, if we go to the world scenario, then pesticide, out of the pesticide which is used 3.69 million tons, 52% is the herbicide use. So ultimately in India, still we are using per hectare, kg per hectare of the pesticide use is very less. It is only 0.24 as compared to China, and the maximum is having United States, which is using 3 kg per hectare of the pesticide per hectare. And coming to the herbicide, out of all the pesticide, if we come to the herbicide, then there are almost 350 herbicide which are registered world over. And these are the 10 important site of actions in which, just a minute, I need to, in which this is a maximum number of active ingredients or registered herbicide are in group 5, which is PS2 inhibitor. Then 58 actives are there in ALS inhibitor, which is group 2, and then followed by our very long chain fatty acid synthesis inhibitor, and then PPO inhibitors. And 26 actives are those which are like their mode of action is still unknown, followed by auxin mimics. So these are the many herbicides are registered world over. This data has been taken from the weedscience.org. But coming to India, if we talk about India only, then we have only 90 herbicides which are registered with the Central Insecticide Board Registration Authority, Faridabad. Coming to the resistance, what is herbicide resistance? This is, as the picture is depicting, this is ability of plant to survive and reproduce. This is both. It should survive and then reproduce. It should set the seed after treatment with a dose of herbicide that would kill the plant. Normally, the plant will be killed with that very dose. But if we designate some plant as herbicide resistant, then that plant is not being killed with the same very dose. And this is following the Darwinian law. What the Darwinian law says that whenever there is strong selection pressure, you are applying it consistently, then the natural evolution theory comes into play. And in case of weeds, this is a reality happening in today's era also. Like when we apply the herbicide, this is one of the stress for the weeds. So when we apply, and there are some weed populations which have different genetic makeup, like among the, if we are talking about, for example, Phalaris minor, then 1 or 2 plants will be there, those who will be having different genetic makeup, which make them resistant. So when we go on applying herbicide, which is like controlling the weed, then ultimately susceptible ones are getting killed. Resistant ones, they are reproducing and ultimately setting seed. Their porosity increases over the time with continuous use. Resistant individuals, they go on increasing in their number. Ultimately, one day we are having the population more than 40% in our field, which is resistant to that very particular herbicide. And we call it that, okay, resistance has been there in our particular weed. Whereas coming to the another topic or another word which is confusing, that is herbicide tolerance. In tolerance, this is also inherent ability of the species to survive and reproduce after the herbicide treatment. But here, there won't be any selection or genetic manipulation to make the plant tolerant. For example, if I'm saying that 2,4-D can't kill Phalaris minor, then Phalaris minor is inherently tolerant to 2,4-D. Same way, I can say that grass killer like FOP herbicides, Cordina-FOP, that won't kill Medicago, or that won't kill Rumex dentatus. So because those are having inherent herbicide tolerance in them. Coming to the world over scenario of the herbicide-resistant weeds, till date, like 537 unique cases of herbicide-resistant weeds have been reported. These maps are being taken from website www.weedscience.org, which is maintained by Dr. Ian Heap in collaboration with the Weed Science Society of America and Herbicide Resistance Action Committee. And they have also prepared this, that, okay, number of resistant species, they are the highest in wheat, followed by maize. Like 86 weeds have been reported evolved resistance against some of the herbicide which are being used conventionally in wheat, followed by the corn, in which like 66 herbicide resistant weed species have been reported by that website, followed by rice, which is like 57. And this slide is depicting the number of weed species which have evolved resistance against a particular herbicide. Like if I'm talking about atrazine, again, this very herbicide, 66 number of weed species have evolved resistance worldwide. Like this is the same source is there, Dr. Ian Heap website. And followed by, this is group 9 herbicide, which is glyphosate. We have 62 weed species. And then others, there are 6 more weed species, 6 more herbicide which are belonging to group 2 herbicide. And then there is a group 1 phenoxaprop ACCS, against which 33 weed species has evolved resistance. Then we have others. Talking about herbicide resistance, then this selection of the herbicide resistant weed population, it begins with the introduction of synthetic herbicide, which in late 1940, we know that after World War II, 2,4-D was introduced for the chemical weed control. There was a revolution from inorganics to organics, we shifted. And then from 1948 to 1965, we have only one case of herbicide-resistant weed, that was Docus carota, which was reported in Ontario against 2,4-D. Resistance was evolved in 1957. The thing is, during this era, insecticide resistance was more pronounced, like more severe. We have only one case against herbicide, but there were like 17 cases per year for the insecticide. So at that time, and we were introducing our herbicide for chemical weed control in the crops, and repeatedly more of the product, like after phenoxyacetic acid, we come up with substituted urea cells, and benzoic acids. So ultimately, like weed scientists were very complacent that no, there won't be any problem in the weeds regarding herbicide resistance problem, because they argued that, okay, as compared to insect, our weeds are like having more population, more progenies, they are longer staying in the field, they don't, like their life cycle is longer than the insects. So they were like very much satisfied that, okay, this won't be a problem with us. But coming to the next era, which is 1965 to 1980, we had 41 cases against like evolution of resistance was, herbicide resistance was reported. And first wake-up call was the resistance which was reported in common ground cell to triazine herbicide in 1968. And it was like later on, it took time to study the mechanism, and it was found that this triazine resistance was due to mutation change in chloroplast PSBA gene, and it was maternally inherited. So the thing was that we were having less cases, but yes, this triazine resistant individual was, we could able to control it with the bentazone, which is another PS2 herbicide. So there was no hue and cry about the resistance phenomena by 1980. Cut to comes the next era, when we had now group 1 and group 2 herbicide being introduced in the market. But moment they were introduced, within 5 years of their introduction, we found several cases of evolution of herbicide resistance in weeds against these very herbicide belonging to group 1 and group 2. And we, the toll rises to 191 cases. Then after 95, when herbicide tolerant crops were introduced, Roundup Ready and Liberty Link, and there are like many other crops were introduced in the developed country. And to the present, we had now 537 unique cases of herbicide resistance weed evolution in the world over. And this is the split up. I have taken this very data from the same website, www.weedscience.org. And there you can see the map where USA has the highest number of unique resistant weed species number, 134, followed by Australia, and then Canada, followed by China. Brazil at the toll is like 50. And we are here, India 5. And yes, Pakistan has 3 and Russia is only 1. So there are many countries, even like South Korea 17, South Africa 17. So like this is not a like new phenomena now. We are encountering this very problem all over the world, and still we are lucky that we are having very low number of such weeds which have evolved resistance against herbicide. So these were the like then the impetus on studying the herbicide resistance. It started with the like in late 90s and 2000, the mechanisms to study, and then how, which factors they are contributing to the evolution of herbicide resistance. It was studied and it was found there are like three main factors: weed factor, herbicide, and cultivation practice. Among the weed, if I'm talking about number one, if there are more number of individual which will be having that biotype or population which is having different genetic makeup, so the chances of like evolution of or selection of the resistant individual will be more. The faster, more the initial frequency, like lolium, it has evolved resistance against 12 or 13 like cytofactions. So more the initial frequency, the earlier or the faster the evolution rate of the evolution of herbicide resistance will be there. Then the selection pressure. If the weed is too much sensitive to a particular herbicide which we use, then ultimately that herbicide is going to put a lot of pressure on susceptible ones, that is killing that very susceptible plants and allowing the resistant ones to produce the seeds, ultimately allowing the or increasing the rate of evolution of herbicide resistance. And then third was the biological fitness. Earlier, when in 1968, triazine resistance was found in Senecio, but then it was observed that that particular plant, after getting that mutation in PSABA gene in chloroplast, that weed was having less seed production, less vigor. So ultimately, that was not able to produce, or the resistance evolution rate was very slow. But with time, we have seen that the weeds which are evolving various resistance mechanisms, they are also biologically fit to produce seed at the same rate as our susceptible ones. And then last is if the weed is having annual behavior, like completing their life cycle within season, or and have high fecundity, means more seed rate, seed production, then it is going to have like faster rate of evolution. And Dr. Jan Heap, they have prepared a one map based on the one study. They found that out of we have around 3,000 type of weed species which are like there present in our ecosystem, causing main problem. So they found that out of those very 3,000 species, weed species, 64% are belonging to perennial category. But when they studied the HR weeds, these 537 herbicide resistant species, and out of those 33% were belonging to annual category. So annuals have more chances of evolving resistance because they complete their life cycle within a very short span, and if their seed production is also higher, plus if they are carrying the resistance phenomena in their dominant genes, so inheritance will be very faster from the one generation to another, rather than if the resistance is coming in the recessive gene. So that were weed factors. And if coming to herbicide factor, more the potency of the herbicide to kill a particular weed, means if it is like killing 90 to 100% of the weed species, the more selection pressure is applying on the weed to survive, and means according to Darwinian law, natural evolution theory. So the resistant individuals, they have more chances to come out more prominently and more producing seed, and ultimately contributing to seed bank. And susceptible ones, they are like their seed are being exhausted with each cropping cycle, but resistant individuals, they are increasing their seed bank with each cropping cycle. So ultimately increasing the rate of evolution. Then the herbicide which are having single site of action or mechanism of action, like ALS or ACCS, they are very particular mechanisms, primary site of action, where the herbicide go and act like for ACCAs, group 1 herbicides, they inhibit the enzyme which are responsible for fatty acid synthesis, or for ALS, they inhibit the enzyme which is responsible for amino acids. So such type of mechanism of action where there is single prominent primary mechanism of action, the faster the rate of evolution will be faster. As compared to some other herbicide like 2,4-D, we have multiple mechanisms, like 2,4-D act by multiple mechanisms by which it produces that last reaction, which is killing of the plant. So it take time, 15 to 20 days for the killing. So they will have less chances or there will be slower evolution. Then if we are using a herbicide, single herbicide year after year without rotation, yes, it will have more selection pressure. And then herbicide which are having longer residual activity, they are persistent in nature. They are going to have pressure on the susceptible ones. They are not allowing susceptibles to grow, but resistant individuals, they are growing and producing seed. So ultimately helping in faster evolution. Then comes the last third factor, which is cultivation practices. We know conservation agriculture or in conservation tillage practices, we rely more on herbicide for weed control. We can't employ tillage, which is a primary mechanism of like we can't have. Dr. Chetan has already told us mechanical weed management tools. But yeah, if we are going for zero tillage, or if we are following the conservation agriculture principle in which the residue cover is there, so yes, we can't go for tillage or any other no tillage. We can't go for any mechanical operation there. So yes, chemical will be more used for the controlling the weeds. So ultimately they are putting more pressure on ecosystem or on the weeds to come out with that resistant individual. And also second, if we are following rice wheat like we are doing in Indo-Gangetic plains, rice wheat monoculture, and there are like monoculture of herbicides also. So ultimately both in collaboration leading to the faster evolution of resistance in weeds. So these are the various decisions which can help in assessing that if we are at risk of evolving resistance. For example, if we are managing our weeds with herbicide mixture or rotation, then we are taking herbicide mixture or rotation with more than 2 site of action. Like one, I'm using microtubule assembly as pre-emergence. Then if any second cohort is there, then I am tackling them with any ACCs or ALS inhibitor, or I'm using some premix containing PS2 and ACCAs or ALS. So ultimately there will be low chances of evolution of resistance in the weeds. And coming to, if I'm using only one, yes, there will be high chances that resistance evolution will be very faster in my cropping system. Then how I'm going for, if I'm using different control methods, then yes, all the weeds will be having under same amount of pressure, and even susceptible one, resistant one, they will be controlled with cultural and mechanical means. And if I'm using only chemical, then yes, there will be higher chances of evolution of resistance in the weeds. Same if I'm using same mode of action, like in rice wheat, I'm using ALS in rice, ALS in wheat, and then that too using many a times in a particular field, like as pre-emergence, early post-emergence, or post-emergence, I'm using the same mode of action. Then yes, it is going to increase the evolution of rate of evolution. Then cropping system, if there is no rotation, there will be high chances of evolution of herbicide resistance in weeds. But if I am following one of the third principle of crop conservation agriculture, then also I can avoid the evolution of resistance in weeds, even in conservation agriculture scenario. Then resistance status, if I am knowing that there is lowlium weeds are there, and yes, ALS and ACCS, they will evolve. So there are high chances that I will get very fast. Like if I'm using chlordinafop or phenoxaprop, and resistance was reported there, and I shift to my another chemical, which is DEN, which is phenoxaden. Though it is belonging to different chemical family, but yeah, this is killing the weed plant with the same mechanism. So ultimately, in 1 or 2 years, the same very weed will also stop being killed with the phenoxaden also. We have seen this in Punjab. Then weed infestation, if it is very high, and we know that our chemical weed control is very much dose specific. there must be some particular dose which should reach the particular site of action and ultimately resulting in weed killing. So if the weed pressure is very much high, then there will be like dilution factor. And if the weed pressure is very low, and ultimately our herbicide will be taken up by the plant in a very suitable or optimum quantity. Then if the control in the last 3 years, we should know that what was my herbicide history and how the control measure was there, how the control efficacy was there. Accordingly, we should decide for the control measure. Then only we can delay the evolution of herbicide resistant in weeds. Coming to the rice wheat cropping system, this is like most prevalent and highly evolved agriculture production system. And it almost in Asia, this is being practiced on 23.5 million hectare area and feed about 4.4 billion people. And out of this very 23.5 million hectare area, we have 57% in South Asian country only, like we have India, Pakistan, Bangladesh, Sri Lanka, Nepal, Afghanistan, Bhutan, and Maldives. And out of these South Asian country, we know that Indo-Gangetic Plain, this constitute about more than 85% of rice wheat cropping system. And because of this Indo-Gangetic Plain, this is highly fertile, and we have seen the year-wise trend of rice and wheat production. For example, in wheat, it has increased from 23 to 70 million tons, and rice from 73 to around 300, like 400. So this was the production which jump was there in Indo-Gangetic Plain, which led to we were able to feed the increasing population. But then there comes the sustainability issues, other than sustainability issue of the water problem, hard pan problem, like soil physical structures, those problems are there. I'm constricting myself to herbicide or weeds aspects only, that because of the monoculture of rice wheat cropping system, both cereal crops, both growing there, and ultimately earlier cropping pattern was different before Green Revolution. After Green Revolution in Indo-Gangetic Plain, rice wheat cropping system was widely adopted, and there was changed weed management practices, irrigation practices, then herbicide use history. So ultimately, predominance of particular weeds keep on changing from one species to another, depending upon if any of these things are being different here or there. Like if we are changing cropping system is different, if it is rice or rice wheat or maize wheat or cotton wheat, accordingly we have different wheat flora there. Water management scenario, if it is puddled or if it is DSR, so accordingly wheat shift will be there. Fertility status, because weeds come according to the fertility status. And then sowing time also played a very significant role in weed shift. As we delay, like if we are changing from wheat to potato or to mustard family, they are being sown in the month of September, October. There will be less population of Phalaris minor in next season. But if we are shifting to wheat and in November sowing, the wheat, we will have more weed problem. So similarly, crop cultivar, if we are using some cultivar which is smothering factor, high vigor, initial vigor is high. So ultimately, this weed, Phalaris minor, this became the predominant weed in the rice wheat cropping system over the season, over the years. And ultimately, this herbicide, to control this very Phalaris minor, we had some chemicals which resulted in shift in weed flora also, which were not being controlled with those were chemicals. Or second, the main problem are herbicide resistance evolution. So we should be able to differentiate by now what is population shift and another what is resistance. Population shift is like if I'm using a herbicide which is effective on weed A and B, over the year, weed A and B, they will be like their intensity, their population are going to decrease, but the weed C, which were not being earlier controlled with that particular herbicide, its population is going to increase over the year. This is called weed shift. But if I am talking about the resistance, then the particular weed was being killed by that very particular herbicide in previous times, but now it has stopped being killed. This is called resistance evolution. So this is the slide taken from that very website. If we go to India, we search it, and then you will find this very table where they have registered year in which particular weed species has been evolved, like reported registers against some very herbicide. Country, we have India. I have narrowed down into India. Then these are the mechanisms of action against which, for example, in 1991, Flaris minor has evolved resistance against PS2, which is like candidate herbicide was isoproteron in wheat crop. In 1994, then Flaris minor, some populations have been reported resistance against ACCS inhibitor, which is like candidate herbicides were diclofop and pinoxidine. Then in 2006, we have multiple resistant population of Flaris minor, which were like resistant to ALS, group 2 herbicide, ACCS, group 1 herbicide, and PS2, which is isoproteron group. So basically, we had multiple resistant population. Then in 2013, we have another Flaris minor population, which was registered to evolve resistance against ALS inhibitor, which are IDO and MISO. Combination, we have Atlantis from Bayer. So then in 2014, another weed was reported, which is Rumex dentatus. It was reported from Haryana, India, and it was reported to be resistance against ALS group 2 herbicides. And then in 2017, 2 weeds from the rice, Cyprus deformis and Echinocloa crusgully, they have evolved resistance against group 2, which is Pisperivac sodium. So this is the overall summary of weeds which has evolved resistance in the rice wheat cropping system. So I have talked about multiple resistance. So what is that? So cross resistance, earlier cross resistance is the one when in a single mechanism, if particular weed has evolved resistance against a herbicide which was used earlier, then that resistance mechanism has allowed that particular weed to evolve resistance against second herbicide of the same family or same site of action to which it was never exposed. So it was like cross resistance. And we have examples. Singh and his co-worker, they found that PS2 inhibitor isoproteron, that was also showing cross resistance to another herbicide which is of different family and different mechanism, which is group 1 ACCS diclofop. And then Kaur and co-worker in 2016, they also reported it from Punjab that codinafop and phenoxaprop resistant Flaris minor biotype were also found to be cross resistant to pinoxidine. So then comes the second terminology, multiple resistant population. What are those, that type of resistance? In this, a particular weed evolved first resistance against one herbicide, then we changed that very herbicide, and in separate selection mechanism, that weed has evolved resistance to second mechanism of action also. So this was classic example. We have found multiple resistant population of Flaris minor in India, where the particular population was resistant to 3 site of actions like PS2, ACCS, and ALS. So like isoproteron, phenoxaprop, codinafop, and then sulfosulfuron. So Shokar and Sharma, they have reported that very multiple resistant population from India. So coming to the history of use of herbicide for controlling Flaris minor, if we see that in India, isoproteron was recommended in 1977 for control of Flaris minor and other weeds. In wheat, it was like broad spectrum herbicide controlling most of the weeds present in the grasses and broadleaf, both in wheat crop. And with the continuous use from 1977 to 90s, like almost 10-15 years of continued use, there was evolution of resistance in Flaris minor against isoproteron, which was like first case of herbicide resistance from India. And it was like first case even in world for Flaris minor. And it was such a like on a larger area and such a severe case that the whole crop of the wheat failed. And ultimately, by 1997-98, many alternate herbicides like codinafop, phenoxaprop, sulfosulfuron, and traloxydim they were recommended. And then in 2009, pinoxidine was recommended for this resistant population, controlling this resistant population. But then what we saw that continuous use of those very herbicide, even for like 3-4 years, like they were sufficient to evolve resistance against codinafop, phenoxaprop, sulfosulfuron, and then multiple resistant population in Flaris minor biotype. They were also reported from other part of the world like Israel, Mexico, and South Africa. So this is basically map showing where the Flaris minor is a main problem, and this is the like northwestern Indian states, Punjab, Haryana, Western UP, where the rice wheat cropping system is being followed, and we have resistant phenomena reported there. When comes this problem, Flaris minor, this problem further deteriorated when in 2013 onwards we found cases of Rheumix dentatus showing poor efficacy, like metsulfuron was not able to control Rheumix dentatus, and then this evolved resistance against, this also exhibited cross resistance against fluorosulfam, paroxylum, idosulfuron, and triasulfuron. Then from Haryana, there were reports that even Amina ludoviciana and then polypozone monspilasis, they also reported to exhibit resistance against ACCAs and ALS, respectively. Then Joker also reported in 2018 that Cyprus deformis, they have evolved resistance against bisparibak sodium and pinoxylum in dry DSR system. And it was also cross resistance to like metsulfuron plus chloriminon combination, then ethoxysulfuron and fluocytosulfuron. Then, yes, from Chhattisgarh and Kerala, DWR scientists, they come up with this report of Cyprus deformis showing resistance against bisparibak sodium. And there were also from southern region of India, there was first case of evolved resistance in Echinocloacris gully to bisparibak sodium. So basically how we'd evolve resistance against herbicide, which like herbicide were able to kill those. So there are basically two mechanisms. One is exclusionary resistance, which is non-target site, and another is target site. This exclusionary resistance is basically here the herbicide is not allowed to reach the site of action in a optimum quantity. So either the resistant individual, they develop some overproduction of waxes or hairiness or drooping habit. So they have such behavior by which their herbicide uptake is now not as equal to the susceptible ones. Or if they at all there is uptake, then their symplast or apoplastic movement is being hindered somewhere. But in some weeds, it has been found that resistance was due to the faster translocation. But in some other weeds, it was reported that the resistance was there due to the slower, there was minimum translocation from the site of uptake to the site of action. Then during the passage from the site of uptake to the site of action, the herbicide on the way, they can get compartmentalized in some oil bodies or peroxisomes. They can get sequestered in like with the other proteins or by metabolites. Basically, they on the way they can get detoxified with P450 monoxidase or other enzymes which are plant defensive mechanisms. They are involved in plant defense mechanism. So they are seeing herbicide as one of the stressors. So they come into play, and various enzymes like oxidase, dismutase. So they come into play, P450, glucoside transferase. So they basically don't allow the herbicide to reach the site of action. So this is the more serious type of resistance phenomena. If we have non-target site resistance phenomena, then it is a like family business. If one weed has non-target site resistance phenomena, then against one particular herbicide, then in time to come, it can evolve resistance against many site of action. It has been observed in USA. Then second mechanism is called target site, where we have two methods by which target site, in one basically the site of action where the herbicide go and bind, that is altered. So their structural changes are there. There is some point mutation which is leading to different protein, and the herbicide is not able to bind to the protein. Ultimately, plant function goes uninterrupted, and ultimately plants survive the herbicide action. Then there can be another site of action overproduction. So sometimes it has been found in glyphosate that there are more copies of ALS and EPSP genes. So ultimately, the herbicide in a particular quantity go to the site of action, bind to the proteins, but there were so many multiple copies of that very gene that plant system goes uninterrupted, and ultimately plants survive the herbicide action. So coming to rice wheat cropping system, various like Dr. Samundar Singh from HAU, they have their PhD on this very mechanism studies, isoprotein resistance, and they observed that this resistance was attributed to enhanced degradation via dealkylation and ring alkyl oxidation. And this was an NADPH cytochrome P450 monooxygenase activated enzymes were there, which resulted in this very degradation of the isoprotein before it reaches the site of action, which is PS2 enzyme. So then in Avena ludoviciana and polypuzone also, it has been found that there in those very resistant individuals, the ALS enzyme that was insensitive. And then in Rumex dentatus, it has been found by my MSc student that we have sequenced ALS gene, and it was found that there was point mutation at 9033 position in ALS gene, which resulted in production of the like resistant individuals. So we have talked about, yes, resistance is there, and yeah, it is a certainty. We have to face it, keeping in view that chemical weed control is the easiest method, simplest method. So we need to like acknowledge this fact that we can only prevent or delay it. We can't like stop it. We have the mechanisms, like we need to aware the farmer. We go on training them that, okay, these are the factors which are responsible for faster evolution. So please go for such techniques that this evolution can be delayed. And then if at all we found some resistant weed population, then we should monitor them and we should tackle them with unified effort. And for that, if herbicide management is to be like guided to the farmers, then first of all, we should guide them about like, don't go for the herbicide to which herbicide has evolved resistance. Like if clodinafop has stopped working on Flaris minor, then straight away go for shift to another site of action. Don't use 2 time or like 1.5 time dose of that very herbicide. Second, go for rotation, alternate herbicide. Like if I want to take one example for Flaris minor, if I know that my Flaris minor is not being killed with clodinafop, then I will first of all, in the next very season, first of all, I will go for pre-emergence herbicide, which is microtubule assembly inhibitor like pendimethalin, or very long chain fatty acid synthesis inhibitor like pyroxasulfone. So these type of herbicide can be used. Then if at all there are like multiple cohorts of Flaris minor coming in our wheat crop, with every irrigation, we are finding another cohort. So to tackle them, if their number is large within the critical period, we have to manage the weeds. So we can go for alternate herbicide, or we can go for herbicide mixture. Herbicide mixture partners should be like the herbicide which are having 2 site of action, like clodinafop and pinoxidine. They are not like perfect partners. If I'm using herbicide mixture, then yes, one partner can be metribuzin, which is PS2, and another partner can be clodinafop or pinoxidine, according to. So that partner, these 2 herbicide, when come together, they will have 2 site of action, ultimately killing the resistant individual. And yes, whatever technique whatever herbicide we use, if you are not using it in a proper way, like 4 hours, right herbicide, right dose, right time, and right method. So without them, we can't achieve successful chemical weed control. So ultimately, basically, we need to follow different tactics in which mechanical operations like tillage, reduced tillage, zero tillage, and then zero tillage with the mulching, which don't allow the weeds to come out. There is a physical barrier. Then various cultural techniques like go for crop rotation if it is possible, and then stale seed bed allow the weed seeds to grow, to germinate after pre-sowing irrigation, and then go for either non-selective herbicide application or shallow tillage, and ultimately go for sowing. The upper 1 centimeter that soil profile will be like first cohort of the weed, which is very much serious, so that can be avoided with stale seed bed. Then cultivar selection should be chosen wisely, that the particular cultivar should be having initial vigor. They should smother the field very early in their crop season. Then planting date can be, I have already talked about it. Then seeding rate can be increased, spacing can be reduced. And last, if after the critical period of crop weight competition and cohorts are coming in your crop, and then you need to adopt any like harvest weed seed control methods, like either don't allow the weed seeds to set the seed, just pluck out seed heads before they they scatter the seeds, or we in Australia, like this is a very common method, or in now in US also, with the more number of resistant cases, they are now shifting to harvest weed seed control tactics, in which Harrington seed destructor or Rhodocop like millers are there along with harvester. They just crush the weed seeds there in the during the threshing operation. So biological, like various allelochemicals have been observed, but the efficacy was observed only in the labs. The same efficacy was not repeated in the field, because allelochemicals, they are mostly aromatic compounds, and maybe in the field their concentration is very much less small. And ultimately also there are two school of thoughts, whether this is crop residue by inherently that physical hindrance, or due to their release of allelochemicals. So yeah, this is like still a researchable issue. Then if we go on applying all the method along with the prevention, from entry of the weed seed into your field, using crop seed which is free from, like recently we had one resolution there, that from the Punjab, Flaris minor seed, if at all they're in the crop seed, how to avoid that how to prevent that dispersal. So basically prevention, mechanical control, cultural control, and chemical control, biological control, when we take them together, then ultimately we are not putting so much selection pressure on one method, and we can control the weeds very effectively. And to this, I think all four partners, pesticide industry, academia, government agencies, should come together with the final stakeholder, which is farmer. So we should all work together. Only then herbicide resistance management strategies can be practically translated into the field. So this is my last slide, where I want to show that, okay, we need to acknowledge this fact that resistance is an evolutionary process, and this is slowly this is coming into our field because of the selection pressure. And we have seen continuous increase in number of resistant species even now in India also. So we should integrate various non-chemical method with chemical method, so that we can confuse the weeds and ultimately reduce the selection pressure. This is all. Thank you, everyone.
Thank you so much, ma'am, for your engaging, elaborated, and eye-opening presentation, ma'am. Now we open floor for the question from the participant. So they can raise their hand, or they can put your question in chat box. So one question already there, ma'am. What is that? Is answered or what?
So I think there is one question: which herbicide are used in stale bed method? So basically, paraquat is recommended there for zero tillage wheat also. Yes, you can use a non-selective paraquat for controlling, and also this is a zero residue herbicide. You can go on seeding like on the same day after 5-6 hours.
Okay, Doga Srilata having some question. So, Gyanendra
may I
ji,
audible,
can you- yes.
sir? May I audible?
Yeah, you are audible.
Yeah, madam. Good afternoon, madam.
Good afternoon.
So my question is, nowadays a lot of this ready mix products are available now, madam. Ready mix combination of herbicides are coming to the market. So from your lecture, it is understood this to reduce the resistance that of broad spectrum weeds. So is it the proper way to use two different kinds in a tank mix product, or else ready mix is effective, madam?
So basically, I was talking about the mixture partners.
Yeah.
So if the both mixture partners, they are having same weed control efficacy against that particular weed,
Okay.
and also both are belonging, or both are like we have three-way mixture also now. So if they are tackling the same very weed with same efficacy, plus they are belonging to different site of action, then that herbicide mixture is a very good strategy to delay the evolution of herbicide resistance, or to tackle the, or to manage the resistant individuals, which are like having target site mechanisms. But if it is non-target site, then yes, it is a researchable issue.
Yeah. Thank you.
Rahul Kumar. Rahul Kumar, you are not audible. Okay. So some questions are there in chat box. Herbicide resistance identification. Is it happening effectively in India?
So which question?
Actually, it was answered already, but it is.
Okay.
I
Okay.
think I thought it was good question, so I have taken again.
Right. Just a minute. I should see it. So where?
Savita Madappa.
Which? No. Still.
It is
pending.
answered, ma'am. It is answered.
So herbicide resistance identification, is it happening effectively in India? Okay, Dr. Geeta has answered earlier. So basically, for example, we Punjab, Haryana people, we are facing more problem than rest part of the India. So yes, there is a lot of awareness among the farmers for the herbicide resistance in Flaris minor. And luckily we have one or two new site of actions also. But yes, farmers are also aware about herbicide use history, and if they are like in every training program, we go on stressing on this very fact that you need to see that if I'm using the same very herbicide over the years, if what particular ACC is like Cordina-4 openoxaden is working in my field, then the next very season I should go for some ALS inhibitor, or some very long chain fatty acid synthesis inhibitor, or like some herbicide mixture partners. So yes, there are methods. But the thing is, money is involved there. We have rapid herbicide testing kits where small plants, 16-17 days old seedlings, we can bring it to lab, and then with the petri plate testing, there you can apply herbicide. That kit is there. You can test it, but the labor is involved. But the best identification methodology should be for the end user. You need to aware them about this problem, and you need to tell them that, okay, this is real happening, and you need to go for it. Otherwise, like USA, we have to shift to cultural or mechanical means, they are resorting to flame feeding, they are resorting to robotic management, they are resorting to mechanical weed control, precision agriculture. They are resorting because there is a need. They don't have such herbicide options available with them, and we are reaching that stage very soon. If not, we are not properly aware about these things.
Nikhilesh Barua having one question. Which herbicide is best to control mimosa found in the crop field?
Which herbicide is best to control?
Mimosa. Mimosa pudica or
mimosa. Mimosa found in crop field. I think mimosa is that a shy plant. So this is a like a broadleaf, but this is more of shrub type. Na? This is having woody structure if allowed to grow. But most probably, I think if it is at herbaceous stage, it are Pixaro herbicide, which is a combination of haloxyfen plus fluorosilum, or we have aclonifan plus diflufenic, and if not the metsulfuron, then dicamba can be tried. If we don't have any 2,4-D dicamba, only tried when there is no herbicide susceptible crops like cotton or like any other broadleaf crop growing in vicinity, because they can reach, they have drift hazards, they are volatile in nature, so they can reach 1 kilometer beyond their point of application. Yes, there are many broadleaf herbicides available with us in wheat, but I don't know mimosa is found in which crop they have there.
Okay, I think they are asking them question from Assam. So
Assam.
they have to mention.
But they haven't mentioned which crop.
Yes.
That is more important. Our herbicide are very much selective. Like herbicide selectivity is based on like which crop to be sprayed, which weed to be sprayed. Both need to be taken into care. Na? So they haven't specified the crop.
Yes. Another question is that herbicide resistance gene is a modified or naturally occurring gene? Okay,
so this is a very like intriguing question. So herbicide are not the mutagenesis. Like this is not like chemical which are spraying, we are spraying on the crop and the crop itself having mutation. Ultimately in nature, mutation is happening already. So the thing is, the herbicide is not causing that mutation. I have told you that initial frequency, selection pressure will only apply if there is one in 1 lakh or 1 in 1 trillion, 1 in 1 billion, that individual, resistant individual is there, which is able to control that very herbicide. Only then the selection pressure will be there. Na? Only then that herbicide is selecting that individual and killing all other. So herbicide is not causing by default, like at the agricultural applied rate, any mutation within the plant system. But yes, there are initial frequencies of resistant individuals varying in the nature itself.
Asir Bachchan Mahapatra having one question, ma'am. Sometimes there is a chance that weed shift can be confused with the development of herbicide resistance in weed biotypes. How to tackle this confusion and confirm the case? So to tackle this confusion, I think we need to monitor our field. Like if I am going into my field and I can judge that I know my herbicide history and I know what is the weed control spectrum of that very particular herbicide, then yes, I can elucidate whether it is weed shift. Like what is weed shift? The particular weed was never being controlled with that very herbicide. What is resistant? The particular herbicide were able to control that very weed. So this is only coming from the experience. If I'm well aware about these facts and I know about my herbicide use history and I know about my cropping history and how the you need to be there in the field to differentiate. Otherwise, while sitting in the labs, yes, this is not at all possible. Okay, Rahul Kumar having some question. So, Ganamji, can you unmute him? Rahul Kumar.
Which one?
Rahul Kumar, he raised his hand. Rahul Kumar.
So I am reading another question. Herbicide resistance or herbicide tolerance, which term should be used? Basically, herbicide tolerance is a good term. Herbicide resistance is a bad term. Herbicide tolerance, we haven't that particular weed was never being controlled with that very particular herbicide. It was naturally tolerant. I took one example where Chinopodium can't be controlled with Codinafop. This is herbicide tolerance. But when Chinopodium is controlling Flaris minor earlier, but now Codinafop is not not able to control Flaris minor. This is called resistance. So herbicide resistance, what weed I am using, this is like relative term. So you need to be very much sure that, okay, this very particular weed was earlier controlled by this very particular herbicide. So if it is not being controlled now, it is herbicide resistance. But herbicide tolerance is some other thing. I think I have made self clear here now.
Yes, ma'am. Tushar Patel also having one question, ma'am. How can monitor and manage the weed population to present the spread of herbicide?
I'm not Tushar Patel. Yes, how can monitor and manage meat population to prevent the spread of herbicide-resistant population? Okay, monitor means you go to field before, like you need to know the herbicide history. You need to know your cropping history, herbicide history for a particular field, so that I can take decision which herbicide to be used this year. Second, then I have to spray that very herbicide with a very right dose, right time, and right method, so that there shouldn't be escape due to poor weed control spray patterns. Like I need to differentiate if it is like in a particular pattern is there. This can be due to the, like that can be due to the spray was not there, or whether those very, if resistance will be there, then that will be very much smoothly scattered in the whole field. And then if I found, and this is seed bank, basically your weed seed bank is now more crowded with the seeds of resistant individuals. That is herbicide resistance now. Susceptible ones, that their seeds are exhausted now, and resistant individuals, they were like filling your reserve. So that is herbicide resistance. Not all 100% weeds are like resistant there in your field. Basically, only some of the like 30-40% when weed seeds, they are like resistant ones. So we say that because that control is unacceptable to farmer or to any person. So we say that, okay, my herbicide has stopped working in that particular field and resistance have been reported there. So we should change the herbicide. So to manage, if I'm using chemical weed control only, then first of all, I need to change that very site of action. I need to rotate the mechanism. If it was earlier ACCS, I need to shift to ALS. If it like you need to rotate it, or you need to go for mixture, or you don't allow that weed to set the seed even, so that the weed seed reserve is not being filled with that very resistant individuals. So these are the monitoring and management measures we can take for tackling herbicide resistant weeds. If it is so simple and so easier, then the number shouldn't be increasing. This is very complex. This is like we are tackling nature babies. Yes, they are there. So yes, we need to manage them by awareness and continuous surveillance, monitoring, management, and joint actions, not one individual. Because if I'm able to control my herbicide resistance in my particular field, combine will come with the weed seeds, which are like resistant individuals. So nobody first of all clean the harvester combines. So they directly come to your field with all the weed seeds. So ultimately, prevention and management, that is a key to manage these resistant populations.
One question is there, at initial stage, how to identify Filaris minor and weed seedling?
I couldn't hear. At initial stage, how to identify Filaris minor and weed seedling. Yeah, this is very simple. I don't know. This comes with experience. Filaris minor, this basal portion is pinkish. You pluck out and it pink sap will ooze out. And then the overall, if I'm looking at the field, Filaris minor plants, seedlings, they will be like lighter in color as compared to your crop seeds. And what I can say, this comes with experience. I also learned while I went to fields. So I can't make you explain here. You can't get it from textbook. Like this is not a textbook knowledge.
One person is asking about resistance factor, ma'am. Please tell about resistance factor.
Resistance factor, sorry, I missed on that part. Resistance factor is basically when we calculate GR50. GR50 is we apply the herbicide at variable doses, at like graded doses. And this is the dose at which 50% of the individuals, their growth reduction is there. So ultimately that GR50 we calculate for all the biotypes or populations we collected from the field. And then we have GR50 for a particular individual. We want to find resistance factor. We should divide GR50 of resistant individual with GR50 of susceptible individual. This will come out to be resistance factor. And more the resistance factor, more will be the resistance severity.
Okay. One question is there from Kavita Sagar. She has raised their hand. So can you unmute her, Kavita?
Which one?
Gyananji. Kavita, you can ask.
Yes, sir. Thank you. Good evening. Ma'am, throughout the session we have been hearing one common word, that is herbicide, herbicide, herbicide. So
Yeah.
I want to know, I'm interested to know from you, which is the apt term, whether it could be herbicide or a weedicide. Because why we use the herbicide in the crop fields? To kill the weeds. Then why could not we use the term weedicide? Rather, we have been always using the term herbicides.
Yeah,
Please,
I
ma'am.
got your question. So earlier when I was a student, it was weedicide. As the science advances, herbicide also, like after 95, we come up with glyphosate and paraquat and glufosinate. Those very herbicides were reported, which were non-selective in nature. They will kill any green foliage or any herb they found their way into. Okay, na? So with time, if I am spraying glyphosate, then this is not a weedicide. This is a herbicide. This will be going to kill every green foliage. If I'm using paraquat or glufosinate or any other selective herbicide, if we use at like higher rate, if we violate the dose, like we use 10 times dose, the same very particular herbicide will behave like killing everything non-selective in nature. So with advancement, just like weed control become weed management, weedicide become herbicide. I hope I have made myself clear here.
Okay.
But ma'am, as just a common understanding that in a crop field, in a particular crop field, apart from, for example, I am growing a wheat in my crop field. So if I find even 1 or 2 population of a jawar, we can consider it as a weed only, because we don't need jawar in the wheat field. So in such cases, is the jawar weed or just a common?
Basically, then comes this is the like basic definition of a weed. You are like classifying there absolute weed or relative weed. Like absolute weed means the plant, basically there is no term like weed now. But I don't want in my particular field, if it is a crop field, then crop only, wheat crop only. If it is a lawn, then grass lawn only. So whatever plant is growing beyond my intended land use, I classify as a weed. And then for a passerby, I am a farmer, my field is there, I am calling that plant as weed. But for you, if you are a morning walker and you will say, wow, what a flower. So the thing is, weed is a very relative term. And then for your question, you are asking that if it is any other crop plant is growing in my particular crop field, yes, that will, for a farmer, yes, that plant shouldn't be there. This is out of place. I will designate that plant as weed. But for others, like for any other individual, there is a like biodiversity. For environmentalists, this is a biodiversity field. So the thing is, weed definition is a very like relative terms. So it changes according to perspective. And sometimes weeds are very good also. For insects, they are pollinating them. They need pollination, everything. So for them, these are the beautiful flowers on which they can have nectar, pollens. So yes, definition varies according to the person to person.
Okay. So Rahul Kumar have one question, sir. Rahul Kumar, can you ask the question?
Maybe the last question.
Rahul Kumar.
You can.
Okay. Yes, you can ask. Okay, ma'am, he has placed his question in chat box. If we can see in the context of escalating multiple resistance such as glyphosate and ALS inhibitors. So what
I'm not
research
finding
back-
where it is written.
Question, ma'am, open question that Rahul Kumar 5.
Rahul
five,
Kumar, please
four.
take my question. लिखा हुआ है बस.
Above, ma'am.
Above. Okay.
Yes.
Okay, I'm going above that.
5.
Yeah, in the context of escalating multiple resistance such as glyphosate and ALS inhibitor, so what research-backed IWM approach have demonstrated success in dealing with resistance development in Bihar? I'm not certain about the Bihar. I can talk about my Punjab, from where I belong to. So yes, herbicide resistance in rice wheat cropping system is very severe here. And we have shifted to happy seeder, smart seeder, or surface seeder, like use of mulch as a physical hindrance to the emergence of Flaris minor, which is like showing resistance against ALS, ACCAs, and PS2. So yes, conservation agriculture is one of the answer there, and we have shifted to it. Also, it is having killing two birds with one stone, that we are tackling our residue problem, we are tackling our crop residue burning, and we are tackling our pernicious weed, which is Flaris minor, which is a satellite weed of crop, and which has evolved resistance against many herbicide. So yes, we are opting. Our research is like on the cultural weed control parameters like stale seed bed or cultivar selection or seed rate. So we are doing that thing in Punjab to tackle the resistant populations.
Okay. And last question from Aniket. Can we make a simple statement that herbicide tolerance in grain is gained by crop and resistance is related to weeds?
Can we make a simple statement? Okay, nine. Herbicide tolerance. I have taken example only of weed for tolerance. For explaining the tolerance, I told you Chenopodium album can't be killed by Coldinafop. Chenopodium is not a crop here, darling. This is a weed. So yes, tolerance, herbicide tolerant crops are there. We have taken a very positive word there to have herbicide tolerant crops when we induce such insensitive genes, which can then ultimately allow the crop to survive the herbicide action. But ultimately, herbicide tolerance and herbicide resistance is with respect to herbicide only. The plant can be any. It can be grown as a commercial crop, or it can be designated as a weed plant.
Okay. So thank you so much, ma'am. On the behalf of organizing committee, I extend our sincere gratitude to ma'am for enriching us with a valuable insight and experience, ma'am. So